Hi there, Professor Jared Rathel. I welcome you to the first part in a two-part lesson, Lesson 3-2. I hope you took the mere 7 minutes and watched the video materials that preceded this lecture. It's a stated clearly video entitled, Can Science Explain the Origin of Life? And in summary, there isn't a complete scientific explanation that fully explains the origin of life as we know it, but researchers in recent years are coming tantalizingly close to actually bridging that gap between abiotic chemical evolution and biological evolution. So, who among you hasn't wondered, how did this thing called life begin? How did order arise from the entropy in the universe? Ordered life in a universe that tends towards chaos. In other words, what came first, the chicken or the egg? Perhaps it was the chicken and the egg simultaneously, as we see in this Suriname toad. Wait, does anybody have trypophobia? An aversion to the sight of irregular clusters of small holes? Well, if you don't know, you'll soon find out.
Yep, parenthood can be a drag. The kids get under your skin and you just want them off your back. Literally. These Suriname sea toads are finally breaking free from mom. And those holes? Those are her back. Her offspring have developed from eggs into frogs underneath her skin. And now it's time for them to move on, leaving her a little cratered. Boy, and you thought stretch marks were bad.
Leaving mom feeling a little cratered? I'm sorry. Little biology humor. Okay, so we spent an entire unit discussing biological evolution. We recognize how new species can arise from existing species through the process of reproductive isolation. And it creates this vast tree of life, just like Darwin sketched in his notebooks. We also recognize that Homo sapiens are just a relatively recent and tiny tip on the mammalian branch. You remember from the little clay video, if you compress the history of life in 24 hours, humans and all of human history, right, we don't show up until 11:58 p.m. But our question today is, what goes here? What goes at the base of that tree? Who is Luca? That's Luca, L-U-C-A, the last universal common ancestor. So the mounting evidence points to chemical evolution creating Luca.
Stated Clearly presents, what is chemical evolution? Scientists have reason to think that the first living cells on Earth came about through a natural process called chemical evolution. What is chemical evolution? How does it work? And how is it different from biological evolution? To answer these questions, we'll start by first dissecting the terms and then look at an example of how chemical evolution can take simple molecules and organize them into complex structured systems similar to those found in living cells. The word evolution simply means change over time. Biological evolution deals with changes in things which are able to reproduce. Living creatures make copies of themselves. The change over time that we see in biological evolution is not just random change. Oftentimes, it is adaptive change. Populations become better able to survive and reproduce within their environments. When conditions are right, biological evolution can even drive a species to develop brand new characteristics and abilities. For this to happen, biological evolution typically requires three conditions: reproduction, variation, and selection. Let's see how this works. Many species of holly have smooth-edged leaves. English holly, however, is covered in spikes which protect the plant from deadly predators. How did these weapons first evolve? Well, when a holly plant reproduces, its offspring often show random variation. They are slightly different from their parents and slightly different from each other. In a forest filled with grazing animals, individual plants which happen to be harder to eat than their siblings are more likely to grow up and have children of their own. Nature, simply by being difficult to survive in, selects who gets to reproduce and pass on their new traits and who does not. In this case, mutations which simply caused the veins of these leaves to extend past their edges gave rise to a brand new weapon. The discovery of biological evolution was an incredible breakthrough in science. It explained how new complex traits and abilities develop naturally in living things. The problem is, biological evolution depends on reproduction in order to work. Reproduction, however, is an extremely complex process in and of itself. This begs the question, how did reproduction first evolve? To try and solve this mystery, many scientists are looking into chemical evolution. Chemical evolution refers to changes in things that need not be capable of reproduction. Examples could be individual molecules or entire chemical systems. A chemical system is a group of molecules that interact with each other. Molecules, structures, and chemical systems almost always evolve or change over time, but they often evolve towards simplicity. Solid iron corrodes into rust when it comes in contact with water. Proteins break down when exposed to too much heat. If simple chemistry is to give rise to something advanced enough to reproduce, there must be situations in which chemical systems can grow in complexity, form new structures, and gain new functions. In order for this to happen, reproduction, which is needed in biological evolution, can be replaced with a much simpler process: repetitive production. On planet Earth and all throughout the universe, powerful natural events take place in regular cycles. The heating and then the cooling of day and night, the repetitive eruptions of volcanic geysers, the rise and then the fall of ocean tides. These events repetitively produce or give birth to new molecules and chemical systems. These products increase over time and often develop new abilities as they interact with their environment. Let's see an example of how this process works. This here is a special molecule called a fatty acid. It's a collection of carbon atoms, hydrogen atoms, and oxygen atoms all stuck together in a specific pattern. Fatty acids are one of many complex molecules that living cells use inside their bodies. They build fatty acids with atoms they get from their environment. Scientists used to think that living cells were the only things able to consistently build fatty acids, but check this out. Lab experiments have shown that if simple common gases, carbon monoxide and hydrogen, are heated up with minerals like those found in the Earth's crust, a variety of complex carbon molecules, including fatty acids, begin to grow. Living cells are not needed. This can happen naturally in underground chambers, heated by the Earth's magma. As pressure builds, these molecules can belch up into pools of water, where a simplified version of natural selection then takes over. Most particles blasted into water will either float or they will sink. Nature selects against them staying in the watery environment. Fatty acids, however, remain suspended in warm water, growing in number as the cycle repeats. When fatty acid concentrations are high enough, they bunch together, automatically self-assembling into a stable ball. This happens because water molecules are attracted to the oxygen heads of fatty acids, sort of like a magnet, but water repels their oily carbon tails. When fatty acids pass near each other, their tails are pushed together by water, eventually forming a ball. As fatty acid collections continue to increase, they join together to make large skins. If fluctuations in these skins happen to make the edges touch, water forces those edges to fuse together. The end result is a stable hollow container, similar to the membrane or skin of a living cell. These containers have a brand new ability. They can trap other molecules inside, acting as an entirely new environment for chemical evolution to continue working within. It's important to note that these membranes do not qualify as living creatures. They can't reproduce on their own the same way living cells do. That said, the development of these membranes, along with many other molecules and chemical systems that scientists have observed, demonstrate an extremely important principle. Chemical evolution can give rise to new characteristics and abilities. Because of this, scientists hypothesize that under the right circumstances, chemical evolution could give rise to systems that are fully capable of reproduction. If they are correct, this would bridge the gap between chemical evolution and biological evolution, demonstrating that chemistry can indeed give rise to life. Scientists at the Center for Chemical Evolution and other research groups around the globe are working hard to test this hypothesis. So, to sum things up, the main difference between chemical evolution and biological evolution is that chemical evolution can produce new characteristics and abilities without depending on reproduction. Because of this, chemical evolution is being investigated as a possible cause for the origin of life.
The process of chemical evolution may have produced the very earliest and simplest cells on planet Earth, proceeding in a sequence of stages, many of which can be replicated in the lab. In the first step, small organic molecules are synthesized abiotically, meaning without life forms producing them. In the video, you witnessed the cartoon rendering of a very real laboratory experiment where fatty acids will self-assemble from carbon monoxide and hydrogen gas in the presence of clay silicates, just like we find in subterranean chambers in the earth. Fatty acids belched into those aquatic environments, like you saw on the cartoon, would be selected for as they float and other molecules sink. Number two, eventually, as those fatty acid concentrations grow from repeated cycles of heating and cooling and heating and cooling, their hydrophilic heads and hydrophobic tails cause them to form into larger macromolecules, large molecules, which again, we see this process occur in the laboratory. In the third step, these macromolecules will form balls and then massive sheets or skins. When the skins fold over onto themselves, you now have a protocell. What's a cell membrane made from today? Do you remember that from 181? It's a phospholipid bilayer. It's two layers of fatty acids. So this protocell is now essentially a primitive cell membrane. With a fatty acid membrane, Even though it's not alive, protocells, they now have an internal environment that's different from the external environment. So different suites of molecules are going to be selectively internalized. Nature would begin to select for or against certain protocells depending upon which molecules they internalized and how these internal environments differed from the external environments. In my mind, the fourth step is the biggest leap. So the self-assembly of some form of replicating molecule, some proto-RNA. However, once RNA has evolved, we are off to the races in the RNA world.
Stated Clearly presents What is the RNA World Hypothesis? If you were to go back in time 120 million years, you'd find yourself in a dinosaur world. 500 million years ago was a world of trilobites and other strange sea creatures. 3.4 billion years ago was the world of the first living cells. And if you were to go back further still, scientists suspect that chains of a chemical called RNA, or something similar to RNA, kickstarted this entire beautiful mess that we call life. RNA is thought to have given rise to life for several reasons. Chains of RNA are found abundantly in all living cells today. RNA is a close chemical cousin to DNA, and with very little help from researchers, RNA chains can replicate, evolve, and interact with their environments. While many details have yet to be worked out, the RNA World Hypothesis is the simple idea that somewhere on our early planet, perhaps in a tide pool or hot spring, the Earth's chemistry was producing random chains of RNA. Once formed, they began replicating, evolving, and competing with each other for survival. As these chains evolved and diversified, some eventually began cooperating to produce the genetic code, a wide array of complex proteins, and even living cells which, from the perspective of RNA, can actually be thought of as houses or survival machines for RNA to live inside. To understand how RNA chains can interact with their environments, replicate, and evolve, we first need to understand the simple process of base pairing. Chains of RNA are made of nucleotides, small molecules that come in 4 different types labeled A, C, U, and G. The backbone atoms of a nucleotide, shown here as a yellow bar, can form strong chemical bonds with the backbone atoms of any other RNA nucleotide. This means that different chains can have completely different sequences from left to right. The parts we call the bases of nucleotides, the colored sections labeled A, C, U, or G, are attracted to other bases sort of like a magnet, but they're selective about who they will stick to. G selectively pairs with C, A selectively pairs with U. When bases find their matches and stick together, we call it base pairing. Researchers have found that with a little bit of assistance, base pairing allows chains of RNA to replicate and evolve. Here's how it works. When a long chain of RNA is suspended in cool water with high concentrations of free nucleotides, the chain can act as a template for its own replication. Nucleotides automatically base pair with their partners on the existing chain. If their backbone atoms form chemical bonds with each other, and by the way, this is the part that currently requires assistance from researchers, we're not yet sure how this would have happened in the wild, a complementary RNA strand is born, one with the exact inverse sequence of the original. If the water is then heated, paired bases lose their grip, allowing both chains to act as templates when the cycle repeats. The great thing about this process is that every other RNA chain produced is a copy of the original, but sometimes mutations slip in. This means that as chains compete for survival and reproduction, true evolution, descent with modification, acted upon by selection, can operate on chains of RNA. As amazing as replication is, base pairing also gives RNA chains a second special ability. When placed in water cool enough for base pairing, but without enough free nucleotides for replication, chains will fold up and base pair with themselves. The end result is a complex shape with certain sticky bases pointing outward because they weren't able to find partners. These sticky, outward-facing bases can cause unique chemical reactions by interacting with other molecules in their environment. A folded chain of RNA capable of guiding a specific chemical reaction is what we call a ribozyme. Some ribozymes break certain molecules apart, others join certain molecules together. A ribozyme's specific function is determined by its specific shape, and its shape is determined by its sequence. If a mutation changes a ribozyme's sequence, the shape can be modified, and so can its function. When ribozymes were first discovered, scientists wondered how difficult it would be for random chains of RNA to evolve legitimate survival functions. Imagine, for example, a ribozyme that could build nucleotides out of molecules it finds in its environment. Across multiple generations, natural selection could promote and refine this ribozyme because the chain would tend to have access to more free nucleotides than its rivals, allowing it to replicate more often. To explore this idea, researchers at Simon Fraser University produced a large group of random RNA chains and examined them to see if any happened to be able to make nucleotides. Surprisingly, some actually could, but they weren't very efficient. Researchers selected out the successful chains and then used a lab technique called PCR to quickly replicate them with slight random mutations. After just 10 rounds of PCR, followed by selection, highly efficient nucleotide-building ribozymes evolved. These are molecules with the lifelike ability to actively participate in their own survival. These ribozymes, and many others produced through similar experiments, are beginning to blur the line between living things and simple chemistry. So to sum things up, the RNA World Hypothesis is the simple idea that the first things to replicate and evolve on our planet may have been chains of RNA or something similar to them. While the basic idea of the RNA world does seem to give us a promising pathway to the origin of life, it's still very much a work in progress. As mentioned, one of several unsolved problems is, how did nature get backbone binding to function without the special enzymes or lab techniques we use today? While many researchers continue to focus on RNA, others are investigating alternative molecules, chemical systems that might replicate and evolve without assistance and could have given rise to RNA. Continual breakthroughs are being found in both avenues of research.
In 1969, an approximately 220-pound meteorite was observed streaking across the sky, falling near Murchison, Victoria, in Australia. The Murchison meteorite, as it would eventually come to be known, was startlingly rich in organic compounds. It contained many of the building blocks required for life. The Murchison meteorite contained a number of amino acids, including glycine, alanine, and glutamic acid. And you remember, the amino acids are the building blocks for proteins, and the proteins, of course, build life forms. Further, the Murchison meteorite contained both purines and pyrimidines. Recall that nucleic acids, RNA and DNA, have these nucleobases where C, T, and U are pyrimidine derivatives, and G and A are purines. So since Murchison, there have been a number of meteorites that have been recovered and analyzed, and they've confirmed that our solar system is teeming with the building blocks of life. It's teeming with amino acids, purines, pyrimidines. And given that the primordial soup of early Earth was bombarded with these meteorites daily, a high-resolution spectroscopy of the Murchison meteorite revealed that there were like 14,000 compounds in this thing. So you've got this soup that's being hit with these meteorites. It creates this chemically complex brew, and then you've got these vast stretches of time. So I'm going to go there in my Walk on the Wild Side video, which you're going to watch as part of this unit. I have one per unit. This one's entitled Exobiology, Are We Alone? And in it, I'll briefly discuss Frank Drake and Drake's equation, as well as Sir Fred Hoyle and his idea about panspermia. Panspermia is this idea that the universe is teeming with life, and that life is vectored, or at least the seeds of life are vectored across the vast oceans of space, hitching a ride aboard meteors. So there's a whole spectrum of variants about panspermia, but the adherents, for the most part, suggests that the self replicating molecules, the RNA, possibly the DNA, the nucleic acids, were vectored by microbes, spores, or perhaps the tardigrades. So, tardigrades, also known as water bears or moss piglets, you can see why they get that name. They're eight-legged, segmented micro animals that live in aquatic environments all over the world, from mountaintops, the deep sea, even in mud volcanoes. We've got these tardigrades in our fish tank in our lab at MON 125. We found them when we go on Protus safari. Tardigrades are some of the most resilient animals known. They're able to survive the most extreme conditions, such as exposure to harsh temperatures from absolute zero to well above the boiling point. They can withstand extreme pressures greater than many times deeper than the deepest ocean trench. They can survive air deprivation, radiation, dehydration, starvation, things that would quickly kill most forms of life. Tardigrades even survived experimental exposure to outer space. So they're capable of suspending all of their metabolic functions for decades. They desiccate and they fragment their DNA, as well as the DNA of the organisms that are on them and around them. And they enter this, what's called a cryptobiotic state. It's called a TUN in the tardigrades, TUN. But these tardigrades, they could be really a perfect vector, capable of moving DNA around planets in our solar system. And they are adorable. Let me show you what I mean.